It's kind of hard to come up with big things after the last few talks, so we're going to go the other way. We're going to get really small and just talk about changing biological systems by rewriting our code. We want to actually predict everything we can from the genetic code so we can rewrite the code to change the phenotypes and create nicer toys, I guess. So we've been doing this along with a whole lot of other people for, a little over 20 years of converting the A's, G's, and T's into the ones and zeros in the computers. I call this digitizing biology. And now, so we have 1 million genomes by 2020. And the challenge is to do what we're describing here, truly interpret the genetic code, in fact, so we can predict outcomes, and so we can start to change outcomes through preventative measures, through selecting the right drugs for you because of your genetic code, and eventually learning how to update the code. The first fundamental experiment was synthetically making this 5,000 base pair molecule, injecting it into E. coli. E. coli read that as regular DNA, made the proteins. Proteins self-assembled. They formed the virus. And once there were a certain number of virus particles in the cell, it killed the cell and spread the virus to infect other cells. So we call this a situation where the software is building its own hardware. All we did was put in a piece of chemical software, and we ended up with these viral particles that now can make trillions of copies of themselves by infecting other cells. The challenge is to understand this linear code because we know in an absolute sense that it does code for everything with cellular life. So that's where we were in 2010. Life is a DNA software system. There's not a species of self-replicating life forms on this planet that aren't DNA software systems. And again, if you change that software, you change the species. This has happened numerous times throughout evolution. Cells have added entire chromosomes, changed what they do. In some cases, cells incorporated an entire other species. That's how we got mitochondria. That's how we got chloroplasts. And they still have their own genomes that replicate separately. So in this effort to design a life from scratch, in my book, I make, what we define as scratch is pretty loosely defined. We're not actually making all the molecules and doing all the work, but we're trying to design the software that does this. We tried to do this on first principles, and we didn't get very far. Life and evolution is very messy, and we liken it to a hard drive that, after too many complex operations, gets very messy. So we're trying to defrag the genome. And to show you how complex this is, and this is an effort led by Dan Gibson at Synthetic Genomics, this is just taking 1/8 of the genome, going from the full cell with about 1,000 genes to about half that or less than a minimal gene, but all the changes to get things back where you can order them by function. And this is really key if we're going to do design. We want to take a block that deals with all the cell replication issues. There's so many different types of energy production. We just want to take modules and decide what you want to use as your energy source and put that in. And using some processes for the first time, we've actually doubled the energy output of a cell and also changed the efficiency of photosynthesis a little bit at the same time. We heard just recently people want to design and have organs just pop up magically or make them mechanically. They have to grow in situ. The environment for our genes and the control is absolutely essential. They don't work well outside of a system. You take any cell out of the body, the genetic code starts to change very rapidly. Every stem cell, every blood cell, this starts to happen with. So doing things in an environment is critical. And it's a complex problem because there's all kinds of levels that go, of response that go into organ rejection. It's not just changing a few surface antigens. We actually have to alter the immune system of the system, and we're dealing with everything from acute injury to chronic immune rejection. Basically, synthetic genomics has to deliver a new engineered humanized pig cells to United Therapeutics. They'll do nuclear transplantation, create new pig cells, new embryos, grow up the pigs, and then test the organs for transplantation. And DARPA's helped us build the first biological teleporter. And there's two simple parts to this. There's sending units, which are basically doing the first part, taking the biological system, reducing it back to ones and zeros. And then the conversion system that goes the other way takes the ones and zeros and converts them back into biology. So you can download an antimicrobial from the internet, or you can download a new vaccine. And so we've actually been working on a number of approaches of things that we can send biology through the internet and download it. SGI has made a simpler version of this. It's an automatic DNA assembly system. It doesn't do the synthesis. We provide that in modules, and it's interesting because it's telling of this time. This is, I think, the first ever biological device where we've had to build in multiple levels of security, so you can't hijack this machine and make Ebola or smallpox or anything else. So we haven't disclosed what all the security systems are, and everything can eventually be hacked, but if you're going to go all that trouble, just buy a kit and do it yourself. But it's available, and they're out in test sites now, and it's pretty amazing. The early versions have make just small pieces, but we've shown the same device is capable of making entire genomes. Or ultimately a box attached to your computer, because it's a matter of, we can send a new vaccine around the globe in a tiny fraction of a second if you have a device to download it and convert it back into something. Basically, pandemics can be stopped before they ever start if we do this in real time and act that way. The other thing, we're looking for things that would be fun to send through the internet to solve problems. Obviously, antibiotic resistance is a huge problem. There's now more people killed in the U.S. from infectious disease than from car crashes, so it's a big change over time. At HLI, we're taking some other unique approaches. We're trying to understand and decode the brain at the genetic level. For example, we have very nice cameras that take 3D photos of your face and generate about 30 million data points that we're using with our machine learning team, trying to get it so we can predict your exact photograph from your genetic code. We're also doing this with voice as well. So everybody's been working on the other end. They've been working at face recognition software. We're working on face prediction software. So we're creating multitask learning using these new approaches, integrating lots of different types of data to try and get to this point where we're taking this data, converting it back to knowledge, where we hope that we can now go back and start reprogramming cells. So stem cells are how we develop all our organs and tissues. We've shown when you, as I mentioned earlier, you take a stem cell or a cell out of the body, convert it into stem cells, the genetic code starts to change when it's out of regulation. So we've had to develop techniques to go back and correct the genetic code just from the errors that develop. But these same techniques are equally applicable to rewriting the genetic code, changing the traits, making the stem cells more youthful, adding other parameters to actually start to do a type of genome engineering that could help us build nicer toys. Thank you very much.